Welcome to this educational session on the basics of image processing. And I will be talking to you about image registration and tracking in particular. So just to sketch the context. So this is a lecture in a series of 3. Last year, Viro Niesen from the Erasmus Medical Center in Rotterdam, he spoke about methods for image segmentation. So how to, using a computer to automatically recognize objects in the images and to outline them. And this year I will be talking about image registration and object tracking, as I told you. And next year there will be a session by Boudewijn Lelyveld from Leiden University Medical Center. And he will be talking about quantitative imaging biomarkers, in particular in neuro and cardiovascular imaging. And before I forget, I would like to first acknowledge a bunch of people, the postdocs and PhD students in my own group, but also other people at Erasmus Medical Center. Viro Niesen, I already mentioned. Stefan Klein is also in our group and does a lot of work on image registration. I used some of his slides. And also the external labs that we collaborate with, especially Boudewijn Lelyveld, who will be giving next year's lecture. So that's just to set the stage. I'm going to talk about, as I said, registration and object tracking, and they're really going to be like 2 parts of this session. And let's see if I can manage to stay within time. It's only an hour or so, I think. So could you start the second presentation, please? The second file. Yeah, the registration. So let's first talk about registration. What is image registration and why do we need it? Yeah, it's secure. Okay. So we're at the World Molecular Imaging Congress here. So we're talking about molecular imaging. What is molecular imaging? Well, most of you probably know. Well, there are probably different definitions, but this is one of them, I guess. It's everything that has to do with visualization, characterization, and quantification of biological processes at the cellular and molecular levels in vivo. Now, there are many different imaging systems that you could use for this. And Roger Chen, Nobel Prize winner in chemistry, I believe, for his work in cloning and making all kinds of derivatives of the fluorescent protein. He gave this overview already 10 years ago. Maybe it should be updated by now, but in which he characterized the different imaging modalities in terms of their spatial properties along this axis and temporal properties along this axis. So each rectangle basically indicates the smallest possible detail that you can image versus the field of view, and the smallest time scales versus the duration over which you can image. Now, this is maybe a very coarse characterization, but what it shows is that the different imaging modalities are of course, complementary. And very often we use multiple scanners to get the information we need to study a particular process. So it means we have images, we have many images these days, images from different scanners, different time points, different views. And this is where a registration comes in, because how do we integrate all this information? So in this drawing, it's really indicated that they serve as different pieces of the puzzle. In studying a particular process, you need different pieces of information and somehow integrate them, different modalities, different time points. So that's where image registration comes in. It's the problem of how to bring 2 or more images into spatial correspondence. Because if you take a patient or a mouse and you scan it with a CT scanner and then an MR scanner, and if it's not the same system, if the patient or the subject is probably in a different pose, or and the images do not correspond one-to-one, pixel by pixel. So that's the problem of registration. Merriam-Webster's dictionary says registration is to make or adjust so as to correspond exactly. So for example, if you have a CT and an MR, as I said, the ultimate goal is you would like these images pixel by pixel to correspond exactly to the same geometrical position in the field of view to be able to, for example, make a fused visualization. And again, these images can be from different scanners, different time points, different channels in microscopy, for example, in spectral imaging, different sections, different viewpoints, etc. So what we're going to talk about in the next couple of minutes is what are the basic ingredients of an image registration method. The first one is what kind of information to use, extrinsic or intrinsic information. The second one is how do we quantify similarity, because the computer needs to compute somehow the degree of similarity between the two images that you're trying to register. Then what kind of transformation to use. It's all about geometrically transforming one image to make it align or match with a reference image. But what kind of transformations do we allow? Should we use? And finally, if we have found the optimal transformation, what kind of interpolations do we use, interpolation methods to perform that final transformation? So we're briefly going to go through each of these steps. First of all, the sources of information. If the two images, you can register many images, but we're just considering the case of two images and we want to match one with the other. If the two images are really very, very different in terms of signal content, the image intensity distributions, then it is very difficult for the computer to tell whether they are aligned or not. And usually in such situations, such as here, with a micro CT scan, I think, of a mouse, and this is some optical image, bioluminescence or fluorescence, it's very hard to tell. And so you really need to use extrinsic information. You need to bring in landmarks, physical objects in the field of view that will light up in both scans. So in this case, you see the dots here and here. They light up in the CT as dark voids, and they light up in the optical image as well. We can use this information to make the correspondence. Either manually, you can just indicate which point connects to which one in the other image. And if you have enough connections, enough landmarks that you can indicate like this, it's very easy to compute from this a global transformation, for example, based only on shifting and rotation to make that image match with the other. And these landmarks can be defined by the user, or maybe they can even be extracted automatically using image segmentation techniques, such as discussed last year by Viro Nissen. So techniques that will automatically pinpoint blobby objects. You can think of specific image filters for that. So this is what usually should be done, I think, if the image contents are very, very different. If they look alike, such as in this completely different application. I'm just throwing in some images as examples from different applications. So this is an electron microscopy image. And I have rotated one of the images with respect to the other. So they're not aligned, clearly. There's a rotation to be solved. But the image content, in terms of intensity distributions, is very similar. If this is the case, you can start comparing intensities per pixel directly. For example, by looking at the joint histogram. So what this histogram shows is for each intensity level in image 1, how many times does any intensity in image 2 co-occur with this intensity. So it's a histogram, two-dimensional histogram, color-coded. Now, and it's very dispersed. It's very broad. But if I now start rotating this second image to make it align with this one, then look what happens with the histogram. it becomes more and more peaked, the intensities start to correspond much better. So this is clearly something you can now start to do some computations to quantify the degree of peakedness of this joint histogram. And we'll get back to that in a second. But it's just to illustrate the effect. So now the question is, how do we quantify similarity? There are different measures being used in the literature and in practice. The most obvious one is cross-correlation, which directly multiplies image intensities. So you have image 1, pixels x, y, so this is just a two-dimensional case. It also works, of course, in 3D or any dimension. You multiply directly the intensities of image 1 with those of image 2 after a transformation t. And the mean intensity values are subtracted, so that if I would add a constant to the intensities in one image, it has no effect. Also, if I multiply the intensities in one of the images with a constant, it has no effect on this measure. So it's robust to this kind of changes in intensity. It's normalized such that the value of this ratio is always between -1 and +1. And since we're really directly multiplying intensities and summing them up, it's a measure that needs to be maximized. You need to have maximal correlation. So again, this only works if the image intensities already correspond, have the same kind of distributions to start with. Another measure that's used by people is the sum of square differences, which simply subtracts the image intensities, in image 1 and image 2, and squares them and sums them. Now, of course, the difference in intensities should be minimized in order to make an optimal match. So this is a measure that needs to be minimized. So depending on how you define similarity, you either need to maximize or minimize this similarity measure. And another one is mutual information, which is always a bit vague, maybe. It's much more a statistical kind of approach. It consists of 3 terms: the entropies of the individual images, where the second one is transformed, minus the joint entropy of the 2 images taken together. And this is also to be maximized. All these measures are only computed for the overlapping regions. And here I give you the definition of entropy, the Shannon entropy, which is the probability of a given intensity. So this is summed over the intensities, the probability of the intensity I times the logarithm of 1 over the probability. And I'll show you in a second why this makes sense. And the joint entropy is the same thing, except that it's defined based on the joint probability distribution. So basically the joint histogram that I showed you, but then normalized. So the challenge here is always to find the best T, the transformation that transforms the pixel positions in the second image to make that second image correspond exactly with the first image. So it means registration is an optimization problem. So here you have the measure, and either it needs to be maximized or to be minimized, depending on which measure you use. In this case, it's a minimization. And just to illustrate, so if they're not aligned, you have, in this case, a higher value for this measure. And when they are perfectly aligned, it's exactly the minimum. And in this case, I allow only shifts, translations, so you have delta x, delta y, but if you allow more degrees of freedom of the transformation, you have a much higher dimensional space where this measure lives. And the problem or the challenge of optimization is to find in this high dimensional space the global minimum of this measure, which is not a trivial problem. This is a whole field of research in itself in mathematics. We're not going to talk about that today, but just to illustrate the point. So let's talk a little bit more about correlation and mutual information. Click, yes. So if I have a CT image and an MR image, I told you when you want to use correlation, you better make sure that the intensity distributions are already more or less alike of the two images that you want to register. That's not really the case here. The bone is very high intensities here. It's very low intensities there, whereas the stuff in the middle, your brain, is grayish in both, although with much more detail here than here. So directly multiplying intensities, which is what you do with correlation, doesn't really make sense, or probably will not give you a good match. But what you could do is transform. Yeah, that's a bit misleading term because we're going to talk about transformations. What I mean is to transform the intensities, so not the geometrical transformation of the image itself, the spatial transformation, but you can apply image filters to enhance certain intensities and suppress others depending on in which structure they are in the image. So for example, if you apply a ridge filter, which enhances elongated structures in the image, this bone structure will light up. If you apply a similar filter to the MR image, also the same structure will light up. So this is just a trick. By pre-processing, by applying the appropriate filters, you can make the images look more alike, so that correlation starts to make more sense. So here you could apply a correlation very well, and it will give you an optimal match. This is just a visualization of the result after registration, where you can show the bone outline on the MR, for example, to show that it really matches. So that's what I wanted to say about correlation. So let's talk a little bit more about mutual information to get a feeling for what this measure does. So again, it's computed in the overlapping regions, because if you start transforming one of the images to make it match with the reference image, there will always be pixels that do not that are outside the field of view of either one of the images. So you only sum over the pixels that are in both that overlap in both images. So again, this measure is to be maximized. It contains, again, the entropies of the individual images and minus the joint entropy. Now, if you want to maximize this measure, you can do this by maximizing these two terms, because they're plus, and or obviously minimizing this third term, which is minus. And again, the E stands for entropy, which is computed as the Shannon entropy. And this is used because it's a quantitative measure of uncertainty. And I'm going to show you that in these examples. So suppose I have an image which has only 10 intensity levels. In practice, you have many more, of course, but just for the illustration, I have an image with 10 intensity levels. And I should have shown the image, actually, but well, suppose this is the, let's say the histogram, but then normalized, so it's the probability distribution of the image intensities in that image. Each intensity has equal probability. So if I ask you to pinpoint any pixel in the image and then guess what the intensity is, you have maximum uncertainty about the answer, because it could be any one of them with equal probability. So indeed, if you compute the entropy, it gives a value of 1. Now, and by the way, this is just a histogram, right, or the probability distribution. So there are many images that can have this distribution. It could either be a completely noisy image with as many pixels having exactly the same any of these values, or it could be. So this says nothing about the spatial clustering, let's say, of pixels with these intensities. So it could either be a completely noisy image, or it could be an image which really has 10 regions that are well clustered. But it just says that there is a lot of uncertainty. There is a lot of structure in the image, and that's the important thing. If now this becomes the the probability distribution. And I then ask you to pinpoint any pixel, random, and guess what the intensity is in that image pixel. You are probably going to give the right answer, or at least with much more probability, if you say 5 or 4 or 6. So there's much less uncertainty about that. And indeed, the entropy gives a lower value. And finally, this is the ultimate case, of course, where only one image intensity level has maximum probability. So if you then select a random pixel, you know exactly beforehand, if you know that this is the distribution, you will know what is the intensity. So zero uncertainty about that. And indeed, the entropy gives a value of 0. So it makes perfect sense. It expresses the degree of uncertainty about the content of the image information. Now, again, if we use this entropy in this measure, what does it mean? The entropy computed for the individual images, for the overlapping regions, needs to be maximized. So we need to go from a situation like this towards that direction, right? Maximize the entropy. And that makes sense because this is simply an image or a region in the image with a constant value. This is completely useless for registration. If I have two patches that are just have a constant value, what is the optimal match? I mean, it can be anywhere. So this is really the background situation, and you want to ignore that. You just want to include or consider or put more weight on the parts in the image that really contain some information, image structure, and that means higher uncertainty. And that's exactly what this thing expresses. But the joint term needs to be minimized. So for the joint term, we have the opposite situation. We really want to go to a situation like this, where this is just a one-dimensional example. So now you should think, when talking about the joint entropy, you should think about this two-dimensional thing that I showed before, the joint histogram. It should be maximally peaked. The correspondence between intensities in one image and the other should be as optimal as possible, as high as possible. So that's intuitively what this measure does. And it works pretty well. The nice thing about it is that since it's computed over distributions, rather than directly multiplying image intensities, there's a much broader set of images and image intensity distributions that can be matched like this. So it's a much more flexible measure that can also be used in multimodality image registration. So talking about transformations, that's another question you have to ask yourself. How many degrees of freedom do I allow in trying to make one image align with the other? If you know beforehand that the transformation is rigid body, so only translations and rotations, then it's a much simpler problem. It's called rigid transformation. So that only includes rotation and translation. If you need more flexibility, including scaling or shearing, it's called, together with translation and rotations together, called affine transformation. And there's still a relatively simple mathematical formula to express affine transformations. But in some cases, you may need much more flexibility, even many more degrees of freedom. And then it becomes really non-rigid, as they call it. So it's look at that. This is like embedded. So it becomes a non-rigid registration includes affine, and it includes rigid. So it becomes richer and richer. So, for example, the way non-rigid registration usually is done to keep things manageable to some degree is they use a grid. And the crossing points, those are like landmark points, and we're going to try to find the local deformation, or basically just the shift in that point to make the corresponding region in one image correspond with the other image. Once the local shift is known for every cross point, and this grid is much coarser than the pixel level, then you can interpolate the shift that is needed for each individual pixel simply by interpolating from the nearest cross points for which the shift was computed. So that's what this is trying to tell you. The new pixel position is computed from the position in the starting point and interpolated from the displacements of the nearest cross points using B-splines. Well, forget about the formula. It's just to show you how this is roughly done, using a coarse grid that is deformed in every cross point, and then you can interpolate for every pixel what should be the transformation. Now, of course, this is very flexible. You can define the resolution, let's say, or the degree of detail, so the distance between the lines, to any desired level of detail. And finally, if the optimal transformation is computed, then we need to apply this transformation to one of the images to make it align with the reference image. And I'm going to show you now briefly what the effects of interpolation are. Interpolating image intensities always results in loss of information, except under very special conditions that are not really met in practice. So believe me, if you start interpolating image intensities, you will throw away information. Whether that's bad or not depends on your application, but I'm just showing here that it's true. So suppose I have an image here. This is from fluorescence microscopy. It's a cell nucleus, and it's from a time series. This is going to come back in a few more slides. And I want to make this second image align with this reference image. Well, you can already guess visually that this is approximately a 90-degree rotation. And 90-degree rotation is multiples of 90 degrees is a very special case. I don't need interpolation for that. I can just flip my pixels, the pixel grid, and I'm fine. There is no need to interpolate there. Is that clear? Yeah. So I can do a 90-degree rotation simply by, for each pixel here, look up the corresponding pixel there, and no problem, no interpolation needed. So I faked this example, of course, just to show you the ground truth. So this is the real rotation of this image without any errors. But if I would use an interpolation, an image intensity interpolation method to do this rotation, and I'm doing this with in steps of 18 degrees, 5 times. That is to blow up the errors a bit. Then you will see what the effects of interpolation are. So I'm using 3 different methods here to show what the effects are. A nearest neighbor interpolation. And I don't know what this looks like in the back of the room, by the way, because your eye also does a bit of blurring. So you should come to the front and look at the details later on if you want. But anyway, if you would look at this very closely, you would see that many of these pixels are out of place. They're really completely messed up to some extent compared to this. So a lot of damage to your image intensity information. If you use linear interpolation, it's really notorious for blurring the image information. So again, probably if you're all the way in the back, all these images look blurred. You don't see the difference, but right here, the difference is striking. It's really... So linear interpolation blurs. If you use cubic spline interpolation, the result is already much better. And the higher the degree of interpolation, the more accurate this interpolation becomes. And you can really reduce loss of information by picking the right interpolation method. So these are the main ingredients that I wanted to talk to you about: the type of information to use, extrinsic, intrinsic, the similarity measure, and then the kind of transformation you need, fine or rigid, and the type of interpolation that you could use. Just to wrap it up, I've made this sort of manual or guidelines, the things to think about, to remember. If you ever run into a registration problem, what should I do? What kind of methods should I use? And I have a software package here that does this and this and this. Is that suitable for the job? So to recap, the first question you have to ask yourself, do these images that I want to align look alike? If they have nearly the same intensities in the corresponding pixels, yes, if this is so, then I can use intensity-based similarity measures like cross-correlation, which directly multiplies intensities, or the sum of squared differences, which directly takes the difference between intensities. If this is so, you could also still use histogram-based methods. If the intensities do not look alike, if there's no clear relation, or if they don't have the same distribution, then you have to ask yourself the question, is there still some kind of relation? For example, in this CT and MR example that I showed you, the bone was white in one and dark in the other, so that's a clear correspondence. And also in other regions in the brain, for example, it was grayish in both. So as long as all the image structures that you're interested in have a clear relation in terms of image intensities, you can still use a histogram-based measure like mutual information. Like I told you before, it's much more flexible than any of these measures that directly compare image intensities per pixel. And if this is not the case, if there's no clear relation, such as what I showed all the way in the beginning, the CT image and some bioluminescence image, for example, completely, I have no idea how to align them, then I really have to use landmarks in your experiments, because there's no other way to tell which structures should be aligned in the images. And then the other thing is think about transformations. Do I really need a fully, maybe you can get from your colleagues an image registration algorithm that does full-blown elastic registration with blah, 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 blah. Do you really need that? The more degrees of freedom you have in your algorithm, the more likely you're going to make mistakes. So it's better to constrain the problem as much as you can. So you really have to think about what kind of transformation do I really need? If you're just looking at the skull in CT and MR, it's going to be a rotation, and I'm fine. I don't need all this full elastic, non-rigid deformation. And then finally, I would recommend to use good quality interpolators, but maybe that's because I've done a lot of research in interpolation and a long time back, and I know what the effects can be. Okay. In wrapping up this part of the talk, I'm going to show you a few applications of registration. Let's see how much time do we have. Okay, so one example I worked on 15 years ago or so is motion correction in digital subtraction angiography. So this is an imaging modality where that aims to visualize the blood vessels using X-ray projection imaging. So this is typically an X-ray image of the head of the patient. The vessels are not really visible here. So they usually use some kind of contrast, iodine or whatever kind of junk, and then the vessels will show up in the image because they create contrast in the X-ray. So still, there's a lot of disturbing image structures. So the trick in digital subtraction angiography is to simply subtract these images. So you make an image before injecting the contrast medium, and you make an image during injection. Well, you probably know the trick. You subtract the images and you get rid of all background except the vessels. That is, if the patient didn't move. Usually, especially when the contrast comes in, there's kind of a shock reaction. The patient moves and you get a misalignment between the two images. And you get these kind of artifacts that may really interfere with the diagnosis or with the assessment of the blood vessels, of diameters, if there's a narrowing stenosis or aneurysm. So this is a clear registration problem. And I developed a method a long time ago to correct for this. It's not so difficult. You can align these images quite well just by rotation and get rid of the artifacts. It's a pretty strong effect. Another application is brain imaging. So if you really want, I was talking about the skull before, the bone. Okay, if that's what you want to match, then rotation is probably fine. But if you really want to were to match the brain of one person, you know, the gray and white stuff, with the brain of another person, or of the same person over time, if you want to study some kind of, you know, developmental parameter or whatever, then you really need a much higher degree of freedom in the transformation. So this is really an example of non-rigid or elastic deformation of one brain to make it match with another brain. Another example is stitching or mosaicing, which is used a lot in certain fields, for example, in electron microscopy. An electron microscope is probably the best instrument you can, with the highest resolution that's available on the planet, if you really want to study things up to molecular level. But the point is, if you zoom in so much on your sample, the field of view is usually very, very, very limited. And if you want to have a complete view of your sample, you need to make multiple scans and then patch them together afterwards. So that's also a registration problem in a way. So you make those scans, you make sure that going from one scan to the next, there's a bit of overlap. There should be sufficient overlap in order to have enough information to perform that registration. And then you can, after performing the registration, you can just stitch these things together and get a complete view of the sample. What else do we have? Oh yeah, this I showed you before, I think. Yeah, that was the example with the 90-degree rotation. So it comes from this movie, which shows the nuclei of a bunch of cells. And suppose you would be interested in studying or looking at what goes on within the nucleus of this cell. It's not so easy if that thing keeps moving around all the time. So what you could do is to register the images only for this part, for this cell, to correct for shifts and rotations, so that you can directly view the what's going on within the cell itself. And a final example is, for example, in studies of tumor, bone tumor progression, if you make scans of multiple subjects, or you have the same subject, a mouse model in this case, and you make scans at different time points, one week after the other, for example, and you would like to study the progress of a tumor, for example. How do you do that? You have scans that are taken maybe 1 or 2 weeks apart, and the mouse was in completely different posture and position. This is clearly a registration problem. It's a bit like this famous, well, you must have done many of these things when you were younger. Find the differences, okay? It's not so easy if they're positioned like this. It's the same with those mice. If you want to look at what happened over time, you need to first bring them into some standard position or something, because then it's much easier, of course, if once they are aligned or in the same kind of posture, it's very easy to see the differences. So that's exactly what my colleagues, Boudewijn Lelyveld, who will be talking next year in this session, what they have worked on. So they combined a couple of mouse atlases, which describe for each bone what it is and what the degrees of freedom are between the bones. So the joints, the degrees of freedom in the joints. So if you know this, if you have this mechanistic view of a mouse, you can use that. So a standardized atlas, you can use that to match every subject that you have, every scan or the bones that were segmented from it. You can use that to make it match with the atlas. So that's what's shown here. So initially they do a global match. You will see that when the movie repeats itself. And after the global matching, they start matching one bone after the other. Okay, let's wait until it repeats itself. So a global registration, and then you see that they're in different postures. So then you start refining by matching the individual bones to the atlas. If you do this for every subject, or for the same subject for different time points, then you have really made a one-to-one correspondence between every scan and some standardized atlas. And then you can make very nice visualizations. For example, you can stretch the bones of any two mice, for example, in any standardized position that you like. And then you can start comparing directly, visually, but also computationally, different parts of the scan. So you start with very different postures, and then you stretch them to a standardized posture. And now let's see if my movie works. Apparently not. Can you click that movie, or just to see if it works? Otherwise, we'll move on to the next one, which is actually more interesting. No? Okay, forget it. I will move on to the next one. Thank you. In the hope that this does work. Yes. Okay, so then you can just click any bone that you're interested in the atlas, and it will show you, for example, for 3 different subjects, for that particular piece, the corresponding image information. And this is really find the differences, make everything correspond, registration problem, and then you can start visualizing or quantifying any differences. So they developed a lot of these tools to do this, to make life easy for experimentalists. Okay, so that concludes the registration part. Maybe